A warm welcome to this platform. Let us discuss growth and development. So, the first thing, let us know what growth is. And this is a viable question. In an exam, you find a question, especially essay questions. I've seen a question where you're asked to define growth, and this question carries 3 marks. So, what do you need to write? So, growth is a permanent increase in size, mass, number of cells, and complexity of an organism. That is what growth is. So if you are given a question, 3 marks, make sure that you don't miss any of the words here. You are assured of the 3 marks. Now, another thing that you should know is that growth in multicellular organisms, because we are going to discuss multicellular organisms, those that have quite a number of cells in the body. So, growth in multicellular organisms involve life cycle. Another term that you need to know, maybe this time around, you define a life cycle. So, what is a life cycle? So a life cycle is just a sequence of stages that an organism passes through during its development from embryonic stage to maturity. That is what we call a life cycle. Now, having defined, or since we know now what growth is, and that growth in multicellular organisms involves life cycle, let us discuss now, or let us center our focus on growth in plants. Now, what do you need to know on this part? So about plants, what you need to know that there are 2 types of growth in plants. As you know, when you put a seedling, the plant of course will emerge and start growing up and also down, also increase in the thickness or the give. Now, 2 types of growth here. We have primary growth. Now, pay attention. So, types of growth in plants, we have primary growth. Now, primary growth, it just involves the increase in length of the shoot, which means the plant, if this is a seedling, it starts just growing, going up, and also going down in terms of the root. That itself is primary growth. So, primary growth is just an increase in the shoot, in the length of a shoot, and the roots. And of course, this primary growth, the importance of primary growth, in the case you're asked of the importance of the primary growth, so it just enables the shoot to reach or to move toward sunlight. It just enables the shoot to grow toward sunlight. Okay, that is the importance of primary growth. Aside from that, it enables the roots to penetrate the ground and of course grow toward water. So primary growth enables the shoot to grow toward light and also the roots to penetrate the ground. So these are the importance of primary growth. Now, another type of growth that occurs in plant is called secondary growth. Now, for secondary growth, it just involves the growth in the width and also the girth. Okay, so the thickness. Here, primary, we are saying is just going up or down, shoot up or root down. But when it starts growing sideways, that is increase in girth, the thickness of the stem. Here, this is what we call secondary growth. So very important to know about this. All right, I guess there's no problem up to this point and you are following. All right, so please, if you've enjoyed so far, if you are enjoying, make sure to subscribe to this channel if you're watching it from YouTube. If you're watching it from TikTok, give it a like. If you're watching it from Facebook, make sure to follow the page. Otherwise, we can proceed. So now, let us look at now the stages of growth. We are saying 2 types of growth in plants. We have primary and secondary. Now, what are the stages of growth in plants? Okay, so there are 3 stages of growth in plants, and under each of these, make sure that you pay particular attention. So what are the 3 stages of growth, or what are the stages of growth? So it all starts with cell division. Now, when you look at cell division or looking at cell division, just look at it dividing of cells. So one cell divided into 2. These 2 cells divide, become 4. The continuous division of cells is what we call cell division. Now, let us formally define it. So cell division, is a process by which daughter cells are produced from already existing cell. This already existing cell, we can say parent cell. So in other words, cell division, we can say, is a process by which daughter cells, identical daughter cells in this case, are produced from already existing cells. In this case, why I've used identical, it is because of a type of division that occurs here, which is mitosis. So we are saying this type of cell division involves or it is involved, the type of cell division involved in this case is called mitosis. Now, what is mitosis? Okay, under genetics also we get to look at this, but here to define mitosis, you should just look at it where one parent cell produces two identical daughter cells, and those cells are genetically, or cells that are genetically identical to the parents. So mitosis is just involved this kind of, or it's a kind of cell division where a parent cell produces two identical, or cell that are identical genetically, two daughter cells that are, or which are genetically identical. That is just mitosis. So very important to know this, that cell division is the type of cell division involved in this case is what we call mitosis. Okay, so very important to look at this. I hope up to this point we are together. Okay, I hope up to this point we are together. Now, what happened during this cell division? Now, during this time, a lot of cells are produced. These cells are identical in terms of shape, size, and also what they contain. The region of cell division is called meristem. So which means where this cell division occurs in the plant, it is called the meristem. Okay. Now, there are different types of meristems, of course, as you know. Or let me just say there are different types of meristem as we are going to look at them. So types of meristem, one, we have what we call apical meristem. Now, when you talk of apical, it's just the end part, okay? The apex, the end part, can be of the shoot or the roots. This brings us to the two types of apical meristem, of which we have the shoot and the root apical meristem. This entails that cell division can occur at the end of the shoot, right at the end of the shoot, or at the end of the root. As you know, can be growing upward or downward. Okay, I guess you've gotten it up to this point. So two types of meristems. Meristem, by the way, some of you might be asking, what is a meristem? So a meristem is just a growth center. So in plants, you find growth center at the end of the root and at the end of the shoot. At the end of the shoot is called shoot apical meristem. At the end of the root is called root apical meristem. Otherwise, that is where cell division occurs. Okay, and we are saying this apical meristem is responsible for primary growth. Okay, so this growth at the tip and the root tip and the shoot is responsible for primary growth. I hope up to this point no one has been lost. All right, let us proceed. So still on primary growth, so we have also, talking of the growth center, we have also what we call the cambio meristem. Now, you know that there are two types of growth, primary and secondary. So primary growth, what is responsible for primary growth is the growth of a shoot and of course the root. But when you talk of cambio meristem, two types of cambio meristem, we have the vascular and of course the cork. And in this case, these are responsible for secondary growth, which is just simply the increase in the width and also the width. Okay. Now, after cells have been divided, we said these cells are the same in size and they are same, or they contain, they are genetically identical. In other words, these cells that are produced now, move to this section of growth, which is the region of cell elongation or cell vacuolation. So cell elongation or cell vacuolation. Vacuolation is, so you can say cell vacuolation. Vacuolation is accurate because of what happened under this part or during this time. So during cell elongation, what happened? So cell elongation is just a process by which cells grows bigger and develop their vacuoles by absorbing a lot of water. So during this process, the cells which divided during the cell division, they are moved to this section of the cell where now they attain vacuoles, okay? And they absorb a lot of water. In other words, they elongate in shape. So they become long in shape, and of course, they attain vacuole. They absorb a lot of water. Okay, so that is what happened during cell vacuolation. So this is just a process by which the cells grows bigger and develop their vacuoles by absorbing a lot of water. Very important to understand this part. All right, and one thing you should note that here, even if there's absorption of water, the cells still remains the same. I mean, even if cell elongate in size, they still the same in shape and of course size. Very important to understand. Now, after cell vacuolation has taken place, it is time now for cell differentiation, or otherwise known as cell specialization. Now, here they specialize. The cells are the same where we are coming from, but when they reach here, they start specializing. And this is because they perform different function. Therefore, they need to undergo differences in terms of the shape, the parts, develop some features that enable them to perform their functions properly. So here, under cell differentiation, we can say this is just a process by which cells become suited for their specific functions by developing specific shapes and undergo specific chemical changes in their cytoplasm. Now, very important here that these cells that are coming from this side, they are identical in shape. I mean, they're identical. Whatever they contain, they are the same shape, size. Now, when they reach here, they attain certain features. And examples of specialized cells that we can find are 1, the collenchyma, the parenchyma, the sclerenchyma, and the xylem, as well as the phloem. So these are some of the examples of the specialized cells. So these cells that we are coming from this side, from the cell division region, the same cells, they move in stages here. They go through cell elongation. The same cells go through cell specialization or differentiation. And of course, they attain those features. And examples of specialized cells are the ones that are just listed here, which are the collenchyma, the parenchyma, the sclerenchyma, the xylem, as well as the phloem. I guess up to this point, we are moving together. Let us have a look at now the region of growth. We said it occurs at a shoot, also at the root apex. So growth at the apices, which is the tip of the roots and the shoot. Some of you, these diagrams, you've seen them in the past papers, and you might be wondering what are the answers to the questions. And at this particular point, you have to make sure that all your questions are answered. After explaining, all your questions should be answered. So this is the tip of the shoot. Now, very important to understand the regions. So the first part here is the region of cell division. So when cell division occurs there at the apex, the cells move now to the second region, which is the cell elongation. After the cells have attained the vacuoles, they are elongating in size and all that, they move downward now where they specialize. So all the processes that I've explained, on top there, you have cell division, just the cells dividing. Then when they are pushed here, the cells grow in size because they've attained vacuum and absorbed a lot of water, but remember they are still identical. These cells need to perform different functions. So they are pushed down here, or they enter the cell or the region of cell specialization, where they now specialize. And of course, I've just explained, or I've just given out a list of specialized cells. So that is what you need to know. So this is the shoot, okay? And these regions, you need to know them, okay? Then coming to the root here. So the root is starting from the bottom. So cell specialization, cell division is on top there, okay? But for the root is at the bottom there. So we have this part here, this cover here is called the root cap. And the function of the root cap is just to protect the root from damage as it penetrates the ground. I've seen a question where this region is labeled, and of course you're asked to give the function. So the root cap is simply protect the root from damage as it penetrates the ground. Now, at the bottom here, after this part, this is the region of cell division. Now, here cells are dividing, increasing in number. Then they move to the second region where they attain vacuoles, absorb a lot of water, elongate, and that is what we call cell elongation or vacuolation. Then from cell vacuolation here right on top, they specialize, okay? They specialize. So that is where you find specialized cells. And some of the specialized cells that we find are the root hair cells, as you can see this. So I've seen one of the question where this part is labeled. What is that part? So this is just a root hair cell. Then, of course, if you're asked of a region, very important that to know that this is the region of cell differentiation or cell specialization. So examples of specialized cells we know, we have the selencyma, the parenchyma, the collenchyma, the xylem, the phloem, all those. Now, very important to understand that when you talk of the vascular system here, the vascular bundle or vascular system makes up the phloem, the xylem, okay, the cambium. So mostly the xylem and the phloem. So this makes up the vascular bundle or vascular system. So when you ask of the vascular system or the vascular bundle, just know that we are looking at the xylem and the phloem. So this is basically what you need to look at in terms of the shoot apical meristem, or where the growth occurs in the shoot and the root. I guess up to this point there's no problem. So please, if you have a challenge up to this point, make sure that you pause a video, you rewind a video, in other words, so that you can really understand. The ultimate goal at the end of is to make everyone understand, everyone benefit. So make sure that you understand this part. All right. Let us proceed now to look at another part of this topic, and this part is called germination. This part is called what? Germination. Now, the question that we answer here is, what is germination? Germination is a process by which the seedlings develop from embryo, okay? The seedling develop from seed embryo. That is germination. So germination is just a process by which the seedling develop from seed embryo. Now, as we discuss germination, what germinates are the seeds. So very important for us to look at seeds before we even go further, okay? So two types of seeds that we are going to look at, the monocot or monocotyledonous seed, and also the dicot. So to start with a monocot, otherwise what we are going to use here is zime or maize, okay? Zime or maize is the one that we are going to use for monocot. Now, mono means one, and cot is a cotyledon. So monocot, it has one cotyledon. Let me give an example of a groundnut. Maybe we can understand it better. A groundnut, you can split it into two pieces, and those two pieces are called cots, okay? Cotyledons. Those are called cotyledons. Because there are two, is called a dicot. But for a maize, you cannot split a maize seed. It has only one cot. or one cotyledon. So for that reason, is called monocot. So in other words, monocot have one cotyledon. Now, what are the structure of the part of the maize seed? So this part, this diagram, very important to know it, and also the parts and their functions. So the first part, which is the outer part, is called the tester. Then, of course, this inside part is called the endosperm, but of course, in this is called the cotyledon. Then we have the cotyledon. No, let me not say that. It has the endosperm and also the cotyledon is right there. Then now this part is called the coreopter. Then we have the plumule. Then we have the radicle. Then we have the coralyza. These are the parts of the seed of a maize or a monocot. A monocot, a typical example that is given is a maize seed. So you need to really know the maize seed. Now, we are saying germination is process by which seedling develop from seed embryo. So which means the one that germinate is the embryo. Now, the embryo is made up of the parts, the coreopter, the plumule, the radicle, and the coralyza. That is for the monocot. So this makes up the embryo. So the parts that makes up the embryo are these that I've ticked. So very important to understand the structure of this seed. Then let us look at the structure of a dicot. Example, we are going to give the example of a bean, even a groundnut, whatever, let me say legumes or a dicot. Now, let us focus on what we have here. This is the bean seed. So it is also, or it has also, this part is called the plumule. Then this whole part is called the radicle. Then we have the opening there, otherwise known as micropyle. Then we have the tester as well. Then we have the cotyledon, which is this. Then the embryo for here, what makes up the embryo is a radicle and a plumule. So these seeds, I've seen them, especially in multiple choice. Who knows this time around? It comes section A, that is paper 2. So let us be expectant. Okay, things are changing. Questions can come from the areas where you don't expect them to come from, or can be phrased from any part of the notes. So very important to understand or to treat each and every information with importance. Now, what is more important here is to look at the functions of the parts. So like we said, the parts here, we have the tester, we have also the tester here. So these parts, some of them are common and they have the same functions. For example, the tester, either in a monocot or dicot, the function of a tester is just to protect the seed against damage and also infection. Okay, so the function of a tester is simply to protect the seed against damage and also against infection. Aside from that, it is also responsible for seed dormants, as we are going to look at seed dormants at some point. Okay, so for seed dormants, which means it prevents the entry of water into the seed. Then we have the plumule. The plumule otherwise develop into the shoot. So the function of the plumule is simply to develop into the shoot. Then of course the radicle, the function is just to develop into the roots. So the plumule develop into the shoot, the radicle develop into the root. So the radicle and the plumule make up what we call the embryo. That is a known fact. Then what about the cotyledon? So in dicot we have, in dicot, or generally here we have the cotyledon and here. So the cotyledon just stores food, and of course it stores food in non-endospermic seeds. So this is, or this has the endosperm. So in monocot, food is stored in the endosperm, but in the dicot, food is stored in the cotyledon. I guess here up to this point, we are all together as we proceed. Okay. Let us have a look at this part now. What are the conditions necessary for germination? What are the conditions necessary for germination? For germination to occur, what are the conditions necessary? So, one of the conditions necessary for germination is oxygen, water, suitable temperature. Now, how do these play an important role in germination? So, oxygen is required for respiration. Okay? Oxygen is required for what? Respiration. And this provides the energy for germination. Then water, which is the moisture also, is important in the following ways. One, soften the tester so that it can split and releases the promule and the radicle. As you know, the promule develop into the shoot, the radicle develop into the what? The root. It also activates the enzyme that provide aqueous medium, otherwise for metabolic reaction. Okay, so that is another function of water. So water is important in the germination for the reasons given. Now, having looked at that, it's very important to look at the types of germination. What are the types of germinations here? So, the types of germinations that we have one, hypogeo germination, and also epigeo germination. So, the first part that you are seeing there is what we call hypogeo germination. So, hypogeo germination, what is this germination? Or where does it occur? So, for hypogeo germination, is as a kind of germination where the cotyledon is, or the cotyledons remains underground. Okay, this is a type of germination where the cotyledons remains what? Remain underground. Otherwise, we can say this is a type of germination where the cotyledon remains underground due to the elongation of epicotyl. So the cotyledon remains underground, and now you have this emerging. As you can see, only the leaf part emerges. This is called hypogeogermination. Okay? Here, when it emerges, the shoot is covered by what we call the coleoptile. So the shoot is covered by what we call the coleoptile, and the coleoptile just protect it from abrasion as it pushes out of the soil. Examples of seeds that undergo hypogeogermination, or let me just put this so that others that might not have gotten it, is as a type, is a type of germination, where cotyledons remains underground due to elongation of the epicotyl. So that is what we call hypogeogermination, and we say it occurs in monocot. So this kind of germination occurs in monocot. Seeds such as maize, such as sorghum, such as millet experience or undergo this type of germination. All right, what about, so now before we even go to the other type, it's very important, this part is a radicle, so the radicle grow into the shoot. Then of course you have the promule. I just wanted to emphasize on that point. What about epigeo germination? What is this kind of germination? What is epigeo germination? So it's as a type of germination. So you can say is a type of germination where cotyledons are pushed, are pushed above the ground. Cotyledons are pushed are pushed or comes out of the ground. That is what we call apigeal gemination. Now, where does it occur? This occurs in dicot. Okay, example, we can talk of groundnuts. We talk of beans. So that is where this kind of gemination occurs. Otherwise, we have this. Then, as you can see, the cotyledon emerges out together with this. You can even see the cotyledons there. So that is apigeal gemination. So these are the types of gemination. So please make sure that you know these parts. As we go on, make sure that you know these parts. Very important. Okay, so please, the other thing that we need to know is on the part that you're going to look at is seed dormance. Before we come to growth in animals, you need to know something about seed dormance, which is just a state or condition during which the seed carries out minimal metabolic reactions. So during this period, the seed cannot germinate. So what you need to know on this part? So you need to know the importance of seed dormance, which is just to give time for seed to reach full maturity, and also to prevent the seed from germinating when the conditions are harsh. Then you need to look at also the causes of seed dormance. So one of the causes of seed dormance is because of the hard tester. So when you have the hard tester, water cannot penetrate, which result into the seed germinating. So hard tester is one of the cause of seed dormance. Then chemical substances such as abscisic acid, okay, keep the seed from germinating. Then, of course, physiological conditions such as the weather pattern can also result into the seed not germinating until the conditions are normal. Then, aside from that, you should be able to know the ways of breaking seed dormance. Okay, so one of them is sacrification or scarification. Okay, where you where you physically destroy the tester. Then, of course, soaking the seed in water, burning the seed. Some seed need to be burnt to break the tester. Then you look at seed viability, which is just the ability of a seed to germinate into a seedling. That is it that you need to know about that part. So at least I've guided on what you need to look at before you go in an exam. Otherwise, we can look at growth in animals now. So if there's no problem up to this point, allow me to move and of course look at this part, which is growth in animals. Now, under growth in animals, we are going to look at the life cycle of a mosquito and of course the life cycle of a housefly. Now, where we are coming from, we defined what a life cycle. I guess you still remember how we defined a life cycle. So we said a life cycle is just a sequence of stages that an organism passes through during its development from the embryo or embryonic stage to maturity. So that is what we are going to simply discuss here. We look at the growth of a mosquito from the first stage until it reaches full maturity. What are the stages that it undertakes? Okay, so very important to look at that part also. Okay, let us look at the life cycle in mosquito. So one thing that you should know is that the life cycle of a mosquito involves complete metamorphosis. So in other words, a mosquito undergo a complete life or a complete metamorphosis. Now, what does it mean, complete metamorphosis? So this one It just involves different stages of the life cycle, or this is just a type of life cycle where different stages of the cycle have different body forms. So the mosquito at different stage has different form. For example, you have eggs which are different from the larva, which are different from the pupa, which are different from, okay? So at different stages, you find different morphological or different body forms. So that is what we call complete metamorphosis. Now, the first stage, or the stages that are involved in the growth of, or in the mosquito cycle, one start with eggs, then now comes to the larva, comes to the pupa, then adult. So 4 stages that we can count here, all right? And we are going to look at what happened at each stage. So these are the viable questions that you expect. And of course, I have a recording on growth in animals. That is the life cycle of a mosquito and also that of a housefly. So questions are answered. You should be able to find them right on this platform if you're watching this from YouTube. Now, the first stage of a mosquito here, let us start from these eggs. Okay, so you have an adult mosquito here. They start from the adult mosquito, which lays eggs in water. Okay, so eggs are laid in water by a female mosquito. A female mosquito is called Anopheles mosquito. So it all starts by a female mosquito after mating lays eggs in water. That is after mating. Then after some time, the eggs hatch into larva. So this next part is called the larval stage. So now, the larval stage, what do you need to know about larva? So for these, they can swim and they can feed. So larvae swim and feed on phytoplankton and of course small organisms such as zooplankton, phytoplankton and zooplankton in other words. And now the larva is the one that develop into the pupa. So what you need to know about the larval stage is that they are active, they can swim, they can feed, and they feed on phytoplankton and zooplankton. These are tiny organisms that are found in water. And we are saying the larva, after some time, changes into the pupa or develop into the pupa. Now, about the pupa, this is less active and no feeding is occurring out of this. I mean from this stage. So this is inactive, no feeding, but it undergo changes inside. Okay, it undergo changes inside, and that result into it developing into into an adult mosquito. So after some time, the adult mosquito emerges out of the pupa. So that is the life cycle of a mosquito. So life cycle of a mosquito all start with the laying of eggs, then eggs develop into the larval stage or the larva develop into the pupa develop into the adult mosquito. So at all stage, you really need to know what is happening. Okay? Now, what is the other thing that we need to know? So we are saying the mosquito that lays eggs is the female mosquito, otherwise known as the Anopheles mosquito. Okay? Now, the female mosquito, you know mosquito is popular for transmitting malaria. So we can talk of malaria a bit. So the female mosquito is called Anopheles mosquito, and of course it transmits malaria. The parasite or the malaria parasite is called Plasmodium falciparum. Okay, so it transmits malaria into or in humans. Now, this mosquito, since it transmits malaria, it is called a biological vector. So a mosquito is called a biological vector. And by the way, a female mosquito is the one that transmits malaria and never a male mosquito. And what is the reason? It is believed that when it has eggs inside, the female mosquito has the urge for blood or thirst for blood. So, in its attempt to suck blood from human, it transmits this parasite. So a mosquito is known as a biological vector. And of course, a vector here is as an organism that transmit parasite from one host to another. That is what we call a vector. So a vector is just an organism that transmit parasite from one host to another. Now, what does it mean in full? What does it mean is a biological vector? Why is it called a biological vector? Because another vector you're going to say is a mechanical vector. But why is it called a biological vector? And this is because it carries the parasite inside the body. And of course, it undergo part of its life cycle in the body of the mosquito. And this is why it is called a biological vector. And I hope we've gotten a thing here. All right, let us look at some of the things, that is control of mosquitoes. How can you control mosquitoes? So mosquitoes, they breed in stagnant water. So one way to control mosquitoes is draining or stagnant. So draining or stagnant water. That is one way to get rid of the mosquito. So draining out of all stagnant water. Very important. Then also spraying stagnant water. So spraying stagnant water with insecticide or oil. When you spray with oil, simply you are covering the water so that no oxygen is absorbed into the water. So you simply suffocate whatever is there. Then also we can talk of biological control. How is this biological control done? So someone should be able to explain how is the biological process done. Then using insecticide can be one way of controlling mosquitoes. Then also clearing bushes around the houses. So those are the ways of controlling mosquitoes. Okay. So some of the questions that you're going to find is ways of eradicating malaria. That is at community level or whatever level. So what are the ways of eradicating malaria? So that is one question you are going to find among the questions that I've recorded. Make sure that you go through that. All right, let us finally look at now a housefly. So this is the life cycle of a housefly. So what do you need to know about the life cycle of a housefly? So also it undergo what we call complete metamorphosis. So we can say undergo complete metamorphosis. Why do we say it undergo complete metamorphosis? So also different stages, organisms, here there are different forms. So different stages, they have different body forms. Okay? So that is very important. Now, let us look at what happens. So this is the adult fly, as you can see. Then, of course, laying eggs. Now, for this, one thing that you should know is that lay eggs, we are saying raise eggs in rotting material. So it lays eggs when there are things that are rotting. That is where it lays eggs. That is after mating. So a female housefly lays egg in rotting materials or in rotting, material. Can be a dead body, can be whatever rotting materials that are there. So after mating, okay, lay eggs in rotting materials after mating. Then after some days, maggots appears. So the eggs are laid. Then after some times, maggot appears. This is a maggot. Now, what are some of the characteristics of maggots? Though this part is called the larval stage, please. So this is the larval stage. This is the egg stage otherwise. Larval stage, just like mosquitoes, these are active. So which means can move. And of course, they move using their lower part of the body, otherwise known as the pads. Okay, so they can move, and of course they can feed. So move, feed. Okay, they can feed by sucking the nutrients. Okay, using their what? Using their pads that is found on the lower part of the body. So they can feed by sucking the nutrients. Okay, they can move. In other words, here they are active. Okay, so they are mobile. In other words, for move, you can say they are mobile. Okay, so after some time now, the maggot develop into the pupa. So just like in the mosquito, the pupa is inactive, no feeding. Okay, then there's just some changes that are occurring inside. Okay, after some time, the housefly emerges. So very important to understand these stages. Okay, very important to understand these stages. What is the importance of houseflies? Some of you might be asking, do houseflies have importance? So the importance of housefly is that they are vectors for pathogens. So they are important for that reason. They are vectors, which means they transmit pathogens such as pathogens of cholera, dysentery, typhoid. Okay, and a housefly is called a mechanical. So a mosquito, I said, it's a biological. You said it's the biological vector, but this one is the mechanical vector. Why? The flies, they carry pathogens on their body. Okay, where it sits, that is where they carry the pathogens and move to another position. It's not from inside. Okay, that's why they are called mechanical what? Mechanical vectors. So they are called mechanical vectors because they carry pathogens on the external surface of the body. What is the way of controlling houseflies? So houseflies can be controlled by sanitary disposal of refuse and feces, spraying with insecticide, use housefly traps, and covering the mouth of latrines in the places where there are latrines, or let me say toilets. So you cover the mouth of the toilets. Okay, so that is what you needed to know about this topic. Actually, we've covered it, everything from the start, and now we've come to the end. I hope you've enjoyed. Please, an emphasis, don't forget to subscribe, and also don't forget to share. Invite some more. Let us all learn together. Otherwise, we can end here. Now, if you are interested to join us for physical lessons, we are found in Kabwata. That is Lusaka. We are found in Lusaka, Kabwata area, just near Kabwata Primary School. If you are coming there or you stay in that locality, you can just get in touch 0976 402563. So the grades that we deal with from grade 7 to 12, otherwise GCE and also IGCSE, all those levels we handle them. But also if you want to register for online lessons, you can still get in touch on the same number. Otherwise, see you in the next one.